Here at the Alan Turing Institute, our mission is to make great leaps in data science and artificial intelligence research in order to change the world for the better.
This podcast explores the research, ideas and technologies behind the data revolution with the people responsible for shaping our future.
Welcome to the Turing Podcast. Hello everyone, and welcome to another episode of the Turing Podcast. I'm your co-host Ed Calstry, and I'm here with the full podcast team today. So I'm going to get them all to introduce themselves one by one so they don't interrupt each other on Zoom, as of course we're still having to record this, being that it's the COVID pandemic year. So B. Costa Gomez. B, how are you?
Hi everyone. I'm doing fine. Hi Ed, and all the other co-hosts that will say hi in a second. I don't want to discriminate everyone. No, everything is fine. How are you doing?
Yeah, not bad. I thought we'd start today by giving ourselves a little bit of an introduction. B, who are you, and how did you end up working on the Turing Podcast?
Yes, give me an existential crisis right before the rest of the podcast. So I'm a PhD student at the University of Manchester, and I was an enrichment student of the Turing in 2019 that got extended until September because there was a pandemic. Newsflash.
And we roped you into the podcast. That's what happened.
Yes. I was invited to be a guest on one of the first episodes. I think it's like single-digit episode, and then I basically got cornered into staying and send help. I'm kidding. No, I was invited to stay as a co-host, and it's been great.
Right, cool. Joe Dungate, how are you? Sorry, I'm saying everyone's full names just so that for the audience's benefit. But how are you doing, Joe?
Yeah, all good with me.
How did you end up being roped into the Turing Podcast?
That's a very good question. I'm still asking myself that. So I'm the communications officer at the Turing, and yeah, thought it seemed like a fun thing to get involved in, and have done a few co-hosting episodes since. Still here, which I think is a good sign.
That is a good sign, yeah. I'm glad you're enjoying it. You haven't left us yet. And introducing our latest member of the team, Rachel Winstanley. Rachel, how are you?
I'm good, thanks.
What's your background? What brought you to the Alan Turing Institute?
So I'm on the digital fast stream, which is the government graduate scheme, and I'm on secondment to the Turing for 6 months, and thought, why not get involved in the podcast? I think various other fast streamers have been at the Turing Institute over the last year and have done various bits of the podcast. I think it's kind of becoming a bit of a tradition that now we just do something.
We definitely should make it a tradition. That's a good point, actually. Just to clarify, you're a civil servant, right?
Yes.
So it's lucky for you that you're one of the people who gets to work here, and instead of working on either COVID or Brexit related stuff at the moment, I suppose.
Yeah, it's quite. I think I have a very relaxing 6 months compared to everyone else. But yeah, I've only been in government for just over a year, and so far we've had an election, Brexit, and COVID, right. And then now we're on Brexit again. So it's been a very dramatic.
It all comes back around.
18 months, yeah.
Yeah, Holy Trinity, Unholy Trinity.
All right, we're gonna start this episode.
Ed, what about you?
That's a good point. I failed to introduce myself. I'm Ed Calstry, and I worked at the Alan Turing Institute for a couple of years as a research data scientist. And I guess I'm going to, make this big claim. It was my idea to set up the podcast for the Institute. And
ooh.
Yeah, I know, bold claim. But fortunately, others also thought it was a good idea and allowed me to do it. So I'm obviously using this as a platform to, promote myself. No, it's been fun, and yeah, I'm enjoying doing the podcast.
Ed influencer. That's what we have.
An influencer, yeah, that's what I'm aspiring to be. Public intellectual. All right. Well, now that I've introduced myself and we've all introduced ourselves, let's get on with today's episode. And we're going to start with, as we did last week for the very first time in the first episode of Series 2, spinning Alan Turing's Wheel of Fun. And Alan Turing's Wheel of Fun, if people who didn't listen to the first episode, what it's going to do is select a game for us to play, an AI-related game. So let's spin the wheel. And the game that's been selected today is Have AI Got News for You? Woo! Exciting. Are you all excited to play?
Yay.
Yeah, okay. That's what I wanted. Yeah, sorry.
You canned laughter.
So the way this game's going to work, it works like part of the TV show, which is where they basically have to complete the headline. So I've got a selection of AI related headlines, and I'm basically going to read them out for you, and you guys are going to have to try and fill in the blanks. I think this is going to be a team effort, but we'll see how this goes. So the first headline is, and this is a headline from New Scientist: Google's AI can keep blank flying for over 300 days in a row. So fill in the blanks. Google's AI can keep blank flying for over 300 days in a row. Give us your best guesses.
Okay. We need to think about things that fly. So, I don't know. I thought it was a drone. My first thought was a drone.
That's a good guess, yeah. Drone.
Aeroplane.
Mm-hmm. You're in the right ballpark, guessing that it's something that can fly, given that it's...
Wow, we really narrowed that one down.
Yeah. I'm going to push you in the direction of something that requires less input to fly. So it does it a bit more of its own accord. Although I suppose in this case it's using an AI to help it, but...
Okay, so... Ooh, maybe it's Google, right? So the weather balloons that they had, that would be nice.
That's a very good guess, and I think I'm going to give you the point for that, B, because the answer is loon balloons. AI can keep loon balloons flying for over 300 days in a row. And loon balloons are apparently a network of stratospheric balloons designed to bring internet connectivity to rural and remote communities worldwide. So, well done, B. Okay, after having said that it was a group effort, I'm now going to change my mind and say, introduce a bit of competition here and give the first point to B. So,
Oh.
No, yeah. we all get a point. This we have equality here. We all get a point.
I want to hear some guesses from Rachel and Joe on the next one. So let's go for it. Okay. So the next article is from the Daily Mail, and this is a 3-part one. So there's 3 blanks in this one, and the headline is AI will blank scientists rather than blank blank, says expert. I'll read it again. AI will blank scientists rather than blank blank, says expert.
Oh, you know what? This one does ring a bell, and I cannot remember it.
Is it will help scientists rather than replace them?
Rachel, I'm absolutely going to give you the point for that. It's not the exact wording, but the answer is AI will empower scientists rather than replacement them, says expert. And
I was hoping one of the blanks would be a curse word.
Yeah, I mean, that went through my brain. I just went, probably not going to say that word.
No, I'm afraid this is a family-friendly podcast, so
yeah,
there are no curse words. So that story's all about the recent DeepMind protein folding story, which in fact we'll be hearing more about later in this episode. But for now, let's go to the next headline to guess. So. Headline number 3, this is from The Times. German university fears AI takeover could blank. German university fears AI takeover could blank.
Rule the world. That's my guess.
Rule the world. Okay.
Stop progress.
Okay. Joe, I'm not quite going to give you that, but you're definitely in the right ballpark there.
Takeover?
No, that's further away.
Colder.
So I tell you what I should have said is that it is 2 words actually. So it's German university fears AI takeover could blank blank.
I should know this. I do a news roundup every week.
Yeah.
What did you say?
Stop learning. No, that's also colder.
Advanced progress?
It's pretty much the opposite of what you just said.
Stop progress. Did I just already say that? I think I already said that. Just repeat it and it will be right.
I think I'm going to give the point to Joe because that's definitely closest. And the answer is German university fears AI takeover could narrow mission.
Yeah.
I was being a bit harsh trying to get the exact wording there because that was never going to happen, was it? So that story was Jacobs University in Germany, and academics have been left shocked by plans for it to be turned into an AI institute by software giants. I mean, AI institute sounds pretty good because that's what we are. But anyway, well, must have been shocking what they heard anyway. Okay, on to number 4. This is from The Independent. Cambridge University launches master's degree in blank use of AI.
Ethical.
Ethical was a good guess.
Yeah, I was going to go for humane or ethical.
Okay, well, in that case, I'm probably going to give you all a point because that's pretty much right. The answer was responsible. So, Cambridge University launches master's degree in responsible use of AI, and that's a qualification designed for tech professionals, which will teach them how to make sure their products do no harm. Okay, so in that case, it's definitely all to play for. It's level playing field here, and this is the final headline. So pay attention. So this one's also from the Times, and this is DeepMind's AI could help save blank as well as the human race. DeepMind's AI could help save blank as well as the human race.
Is blank just one word?
It is indeed one word. I'm going to read it again because I may have missed out one slight connecting word there. DeepMind's AI could help save the blank as well as the human race.
World.
Planet.
Environment?
Yeah, I think in fairness, I'm going to have to give it to all of you. The answer was planet. So well done. And again, this is another story about one of the topics we're going to be talking about on the podcast today, which is DeepMind's success in the Cas protein folding challenge. So stay tuned to hear more about that. But for now, thanks to my podcast hosts for participating in Alan Turing's Wheel of Fun. How do we think that went today?
Thank you for organizing the topics for us, and I think it was quite nice. Maybe we should make it a competition next time.
Yeah. Fight it out.
But it sort of was. You all. It was a draw today. So well done, everyone. Imagine.
I don't like drawing.
Sounds.
I like winning.
Yeah, I think I perform better when there's a competition.
That's fighting talk. Okay. Well, we'll have to ramp it up for the next episode then, and include a slightly more competitive edge in whichever game that the Wheel of Fun chooses for us. On that note, let's go to the interview. Hello everyone. Today we're joined by Professor Tim Hubbard, who is the head of the Department of Medical and Molecular Genetics at King's College London and Associate Director of Health Data Research UK in London, as well as being the Head of Genome Analysis at Genomics England. Tim, welcome to the podcast.
Thanks very much. Nice to be here.
Yeah, well, welcome, and thank you so much for joining us. I know that this is a bit out of your usual
comfort zone. But can we start? Can we start by getting you to give us a bit of your, potted bio so that people listening to us can know who you are? Like, we'll talk a bit later about what's going on Genomics England and HDR UK, which is Health Data Research UK. But what was your involvement in the original Human Genome Project, and what brought you to where you are today?
So I was actually a structural biologist for quite a few years, working on prediction problems and structural databases. But I was looking for a group leader position, and I was in Cambridge, and Sanger Institute was advertising. And, so I got recruited, and they said, but maybe you'd like this slightly more senior position. And it would just involve looking after the analysis of the human genome, and it won't take up any of your time. And I never did structural biology ever since then.
I was going to say, it's famous last words.
Exactly. That was 97. And of course, that was just before things really got scaled up. And so, so we sequenced a third of the human genome at Sanger, had the draft sequence in 2000, publication in 2001. And then a lot of work on analysis, and particularly the GenCode project, which is working out where all the human genes are. And that project's still going on at the European Bioinformatics Institute. It moved from Sanger, and it is the reference gene set pretty much worldwide.
It was a big project that I'm sure many of our listeners will have heard of, at least in passing, the Human Genome Project. And I was going to ask you, but that you did mention how long it was. 2000, right?
That's 2000, yeah.
Yeah, so it's a long time ago for someone like me, but I won't embarrass you or any of our older listeners. But yeah, it seems like a long time ago. The world of biology has changed a lot since then, I'd say. B and I have both, although we're at Turing, it's an AI and Data Science Institute. We're both biologists by training as well, so we're very familiar with all of that. But before we dive into some of the other stuff that you've been working on more recently, can you just expand a bit? What was the purpose of the Human Genome Project, and how did it pave the way for the modern world of biology we're in now?
So this is really genomics, that every organism has a genome, and that's essentially the blueprint of how that organism is put together. It's DNA. It contains, the genes, and those are read and turned into proteins and, manufacture everything in the cell. It was the scale up of sequencing that enabled us to start sequencing whole genomes, progressively larger and larger ones, culminating in the human genome in 2000. But actually, there was another change which happened a few years after that, which was the change in sequencing technology, because the first human genome was very, very expensive. And in fact, at the time, we didn't think we'd be able to sequence that many types of genomes because of the cost. Then next generation sequencing came along. This is basically massively parallel. Another technology that was developed in the UK, in Cambridge, although it was bought by Illumina in the States. But essentially the price dropped 1 million-fold, and the speed, you had thousands of people working around the world sequencing the first human genome. It's now a run on a single machine for about 27 hours to sequence your genome, any individual's genome. And so that's meant not only can we start sequencing lots of humans and in the health service, and that's what Genomics England's doing, but you can also sequence anything. Any organism. The Sanger Institute's trying to sequence, all of life in the UK, different organisms. And you can do investigations at the level of what's going on in this cell right now, individual single cell sequencing, simply because it's just so cheap to sequence everything in a particular sample. So that's what's really changed things. Actually, it's the price drop. And that's led to big data biology, really.
I was just going to add that it was not just biology that got changed. It affected a lot of people's lives and things by, for example, DNA analysis of people that have committed crimes and things in comparison. And so that has allowed, innocent people from being, released or convicted and stuff. So just an idea of how impactful. Yeah. So with the sequencing, there was a possibility to compare. And so you start having a lot more.
Right. You also have the rapid sequencers nowadays, with sequencing like the COVID virus genome, and it was a big thing in Ebola as well, the outbreak. So the technology's come a long way, and it puts Moore's law, for listeners who are familiar with that, it puts it to shame really, because I think Moore's law is transistor size or halves every
computing power, I think.
Computing power goes up every unit. Well, it's exponential, but
doubles every 18 months, roughly, I think was the number. But this
Fantastic.
Is way above
exactly.
this.
Yes.
I mean, basically, I think as a technology, it's the biggest change, drop in cost and speed up of any technology. Yeah.
You've made the point that I was trying to make, better than I would have done. So thanks for that. I mean, I was biology undergraduate about 10 years ago, and the next generation sequencing was just becoming a big thing then, I think. But yeah, we're in a different world now. Whilst we're on the topic of structural biology and the developments that have been made, there's a really big news topic that I think we need to discuss, which is the recent breakthrough of AlphaFold, DeepMind's solution to the protein structure prediction challenge. So, Tim, can you tell us a bit why this particular challenge has been considered so important to solve, and why DeepMind's result has been in the news in the last few weeks?
So, this is what I was working on before I got into genomics, really, and a gene is a protein. A gene codes for a protein. It's a linear sequence of amino acids. We've known how to convert between base 4, which is the genome, to base 20, which is 20 amino acids making up protein sequences. And we've known for 50 years that these strings fold themselves up into a unique three-dimensional shape depending on their sequence. So you look at that and you think, well, you should be able to work out how that happens, what the shape should be, but it's turned out to be very difficult. So difficult, in fact, that there's been a competition running every 2 years for the last 25 years called CASP, which basically evaluates methods in a blind test setting. And I was one of the organizers for that for the first 10 years, pretty much. And so there's been progressive progress, but it was certainly far from solved until this year. So DeepMind started competing the previous CASP in 2018, and they beat everybody else, but they certainly didn't solve the problem. You were getting rough structures. But this time round, they've produced most of their predictions are almost indistinguishable from experiment. So, good enough. I mean, so good that you can use them for investigating drug interactions, that kind of thing. So there's still some corner cases in this, but rather like having the first human genome, I think we can end up having the first structural proteome, where we've got all the structures of the human genome and any other organism, rather than just the few we have now. It's probably around, I don't know, I looked it up, it's about 18% of the human genome of the sequences of proteins that actually have a structure right now. And that's despite massive investments around the world doing large-scale structure determination. Structure determination is very difficult. It takes years sometimes, trying to find a way of crystallizing, purifying the protein, various other techniques more recently. There are very large structures which are even more difficult. So this looks like you'll be able to do it systematically. So it's really a big, big event. And I think you can look at, people look for, we have the human genome. You can, now we have the blueprint. Well, actually, we didn't quite have the blueprint. We had the blueprint, but it was encrypted because you couldn't decode what these proteins look like, because the shape they have determines what they do. And so if you can't get the shape, you can't tell what they're doing. You can't tell which other proteins they interact with. Essentially, you can't build a digital model of how a cell works. And I think by having this new set of this translation, it's a bit like a Rosetta Stone, then you can potentially build on that. You can potentially work out all the interactions, build a digital cell at the mechanistic level. And that will massively advance biology as a whole.
Thank you for answering. It's just, for the next question, actually, it was inspired because, as Ed mentioned, we have a biological background, but I actually still work in a biology group. And when this came out, I actually talked to my group. And so this is questions based, try not to go too deep into the biology and try not to be too sciencey, but I couldn't let, the opportunity pass without passing along the, the questions. But what I guess biologists would like to know is, how sure can we be that AlphaFold's predictions for the structure of a protein that hasn't already been verified experimentally is really correct? And do you think that the competition has a sufficiently diverse set of protein structures so that the validation can be actually expanded and generalized?
So one of the things, this business about the reliability of a prediction is important. And certainly in the past, I mean, I kind of used to summarize this as, well, people can predict, roughly the right fold, maybe 50, 60% of the time. The problem is they didn't know which 50% were right. This isn't true this time round. Here, the AI, the network, not only does it produce a three-dimensional structure at very high resolution, it produces a confidence for every residue in the structure. And so, you get an indication of where it's confident. I mean, basically it's confident, but if it's not confident, then you get a low score. And so, the confidence. And the CASP competition these days is around 100 targets. So it's pretty diverse, in terms of, and it's not just small proteins. I mean, there's some very big proteins in Cas14. So I think it's pretty universal. It doesn't seem to have areas that it can't address. I mean, there are always going to be corner cases. There are some proteins where it's clear that there are chaperones involved in folding. Certain situations, we don't really understand what the boundaries of that is. So there will be biological exceptions, I'm sure. And there are things like some parts of proteins are disordered. They don't seem to have a structure. Now, I think the assumption is that quite a lot of those cases, there's some sort of induced structure when they interact with something else. Now, I'm guessing that this AI algorithm can probably, with some tweaks and evolution, will be able to address that sort of thing too. But I think the other thing to say is that this is an algorithm they've developed, but DeepMind themselves say, this is a proof of what you can do with AI. And Demis, the CEO of DeepMind, said in a talk at the CASP meeting last week, he didn't think this was necessarily the only way of solving the folding problem with AI. So there's a whole community there. From 2 years ago, they started using different AI methods. They did better than this time around than DeepMind did 2 years ago. So I think that now there's basically the community's been pointed in the right direction, and there's going to be evolution of techniques.
Presumably the competition is ongoing, and there'll be another entry from DeepMind and others next year, I imagine.
Yep, I think that's correct. And there's still areas, for example, RNA structure we need to know these. I mean, RNA in some ways it's a simpler problem, or should be, but then there's modifications of RNA. It's not that straightforward. We're going to need to know all these molecules if we're going to fit all these pieces together to make up an entire cell. And if you think of COVID, for example, it's a small genome, but it's got a small number of kind of little adapter molecules. And there was some part of the CASP competition was predicting those. And where do they fit? Where do they kind of interfere with different parts of the cellular system? They're exactly where they fit with, that's a potential thing to do if you can work out the interactions.
Yeah, I think it's interesting you mentioned the potential application to COVID-related stuff. I think that's what's missing from a lot of the way the media is reporting the story at the moment, is that it's like, wow, this amazing breakthrough. Quite difficult thing to explain to the public why protein folding is an interesting or important topic. I think the key thing to focus on is, what are the potential applications of this? So just to recap, so as we described at the beginning, like, we've been in the sort of era of genomics for the last 20 years since the Human Genome Project. Nowadays, it's really easy and cheap to sequence genes, which is just lengths of DNA. But as you described, that's only really the encrypted blueprint for what's really going on in the cell, which is all of the proteins. And so now, from those DNA sequences, we already can work out the protein sequence very easily, but we can't work out very easily the protein structure without experimentally looking at it with X-ray crystallography. And that's where this competition's come in to find a computational way of doing that. And the news story there, therefore, is that DeepMind have found a really good computational, or in scare quotes, AI way of doing that. And that sounds amazing, but what are the potential benefits of that? How could it revolutionize biology further?
So I think that probably takes me to Genomics England, because there the whole idea is whole genome sequencing's got really cheap, and it's actually realistically to use it as part of clinical care. Now, what can you do with that? Problem is, we don't understand the genome very well, partly because of this problem of protein folding. But there is a subset of conditions, rare diseases and cancers, where you're looking for a single change, in the case of rare disease, that might explain a condition. And so Genomics England, which did the 100,000 Genomes project in the NHS and is now becoming a permanent entity because it's morphing into the Genome Medicine Service as now. So there will be 500,000 people sequenced for clinical care in the next 5 years. But it's targeted on certain conditions. That people are not being sequenced to predict what's wrong with them. It's the case of, if you are ill with one of these conditions, and we haven't got a diagnosis for you, then you're eligible to be sequenced, because there's more chance of us getting a diagnosis. Now, still, after doing a whole genome, we still can't interpret most of those cases. So there's a huge number where, there's basically every genome has around 4 million differences out of 3 billion. Which of those 4 million explains the condition? Once you start having this protein set, you can start mapping those variations onto all those protein structures. And that's likely to improve our ability to interpret what might be causing the condition, particularly as you start building models where you have all the proteins and you know how they all interact with each other. So that's the difference. It's going to improve kind of mechanistic explanation of why this mutation actually is having an effect on this person, and maybe not in this other person who's got some other mutations around that maybe are compensating.
So this is so interesting. I was just thinking that because there are so many diseases that are directly related to protein misfolding or to protein interactions, that this can be a way of studying those diseases in a simpler way. Like, if you can produce the structure, then I guess you can simulate all of the things that can happen, and how does that influence the folding, I suppose.
Yes, there was an attempt to have a project, a big sort of massive EU project called, digital twins, the idea that we might get to a point where we have a digital version of ourselves, and that's where you do your analysis to interpret what, might be the best treatment for that individual in a much more complicated way than we do at the moment. And it depends on us having much better understanding of how biological systems work. But I think that the sort of, the block to that has been removed now, and we can now make it's a potential jump. It was a jump having the human genome. It was a huge jump being able to collect data with next generation sequencing. This is unblocking another bit of science that was, limiting what we could do. And of course, it takes us into a big data world with AI, where it's clear that AI can do things just as it can play better chess and better Go. It can also do things that, in this case, where we really had not solved this problem, despite the collective effort of scientists around the world for 50 years.
Well, I must say that up until now, I mean, it depends what counts as AI, but certainly a lot of people in the public will have seen things that Google and by extension DeepMind are doing as, that's what AI is at the moment. And the examples up until now had been pretty cool, but they'd been games, so they hadn't been real world problems. They'd been, making a chess AI, making a Go AI, making a StarCraft AI. Those are the examples that I've heard of. But
what do you mean that's not a real problem?
Fair enough. These are important things to solve. Well, actually, you could argue, I mean, they're taking the fun out of it, whereas protein folding, like, people don't want to do that. They just want to get on and solve the disease, right? So
well, actually, there has been a game around protein folding where people have been playing games which in fact Demis said inspired him partly around getting interested in this. And also, he said also the fact that there was a framework for testing who's made progress was a key thing as well. I mean, the CASP competition, this idea of having an evaluation framework, it's kind of part of reproducible science. How do you measure if algorithms are working and assess them against each other? It's quite a big effort, actually. I mean, it's had a lot of funding from the NIH. There's around 60,000 predictions every CASP every 2 years. And of course, they all have to be evaluated. So quite a large industrial process handling the predictions.
Well, when you say evaluated, so at the moment, the training data, I suppose, that the AI is using is the protein structures that have already been evaluated by these manual methods of X-ray crystallography or whatever else they use. But I guess one of the things we sort of picked up on earlier, which you've just highlighted there, is there, like, even if the software version, even if DeepMind's AI can now predict a protein structure to, a high likelihood of being correct, it's still going to have to be experimentally verified in any sort of use case, like if it goes into a medicine or something.
Well, I don't think so. I think we're going to get to a point where it's clear proteins fold by themselves. It's clear that there's something in the chemistry and physics of the molecules that allow that to happen. And it's not perfectly pure, the method they have right now, because it does rely on what's called multiple sequence alignments to some extent. It's not quite, this only was announced last week, so we haven't got some of the details of the algorithm. But a multiple sequence alignment, it's not just the sequence in that particular cell, it's other related sequences in other organisms. So, of course, bringing that extra information in some ways you're cheating. You're using information that wasn't accessible to the actual process that's happening as the thing folds. But I'm pretty sure we'll get to the point where we can make a prediction as good from the individual sequence as well. It's just a question of time. And once you can do that, then you can start interpreting any variant in that sequence and work out if it's going to stop the folding, for example. And of course, there's folding. We know that, things like dementia, Alzheimer's, they seem to be linked to misfolded proteins. So these, there's applications everywhere, but, you can point to different, medical problems, biological problems where folding is a particular issue.
So that's interesting that there are clearly diseases which are known to be protein misfolding problems and so forth. Understanding how protein folding actually works is definitely going to give people ideas on how to solve those problems. I was really interested by a phrase you used earlier, which I think you were talking about some EU projects. Used the words digital twins, and at the Alan Turing Institute, we've got a lot of projects which go under that name, but but rather than being biology related, they're engineering related problems. So we had one podcast episode earlier talking about one of these, and it was creating a digital twin of like a steel bridge. The digital twin was the computational version of it, basically a computer model. But it was related to the real one because it had, input from the sensors on the real physical bridge. But what it sounded like you were saying is that something almost comparable for that is envisioned when we get further down the line of really understanding how different proteins interact in cells by building a digital twin of a real person or a real cell. So is that something that you see as being like a milestone that will be reached in the next decade or something? Or
I think it depends on what level you do it at. We remember a human being, it's about 37 trillion cells.
Right.
All of those cells, we don't know how many cell types there are. We've only really understood how important epigenetics is in the last, 10, 15 years. The ENCODE project was something that came after the Human Genome Project in trying to annotate the genome and seeing how, the DNA isn't any different, but the modifications of the wrapping in the cell packaging are different. And there are modifications of methylation, and those kind of fix a cell type's properties. So, a blood cell knows it's a blood cell and keeps on making more blood cells that are the same without having this expensive regulation structure. So we're a long way off building a model which can even model at the sort of molecular level, even simple things. I mean, we can't actually work out where the genes are directly. We do that by collecting external data and mapping it onto the genome, because we can't build a system of all the proteins that are involved in the process of finding the start of a gene and splicing and everything else. Those are big machinery, and we haven't been able to put that together. But there's a chance we can start on that process now. So I think some of the cellular systems, we're going to start being able to build really molecular models, and a lot of understanding will come out of that, because a lot of diseases are molecular defects.
That's really interesting. I just want to make a point for our potentially non-scientist listeners that what we're always doing in science is creating models. So the point you made about the level of detail is really important, because we can't have the maximum level of detail, which is to the atomic or subatomic level, and understand everything, because that would be simulating the entire universe, and that's far too hard, maybe impossible. But the point is, you don't have to have a model that's at that level of granularity, that detailed, to have interesting and informative insights into how the real world actually works. And in this case, how, people's cells actually work in a way that's informative to medicine.
And so that really takes you to the kind of Health Data Research UK world, where humans are very complicated. We have a large number of cohorts, research cohorts, where people have volunteered to, be examined, have their data collected. But that probably is not enough data. So what you'd ideally like to do is to do that over whole health systems. Look at all, because, volunteers, that's going to be a certain subset of people that are prepared to volunteer, rather than everybody in the population. The problem then, of course, is privacy of data, because volunteers are one thing, the whole population, they're concerned about their data, and we've seen that with various issues in the UK around data privacy. And so actually one of the things at HDR UK is, can you construct a framework to make it easier for researchers to discover what data sets there are and to securely run algorithms on that data in what we're calling trusted research environments, where data doesn't move, it stays there. And you bring the algorithms to the data, and the only thing that goes out are summaries, which aren't identifiable to anybody, but are, research results. And so you have to do that for genomics because there's nothing you can do to anonymize a genome. A genome, you need every bit of information to try and interpret it. So Genomics England has built such a thing, and that's what we use for doing research analysis on top of all those genomes, partly to try and explain the ones that we didn't get interpretations for, but also to enhance understanding. And there's around 1,000 researchers actively logging into that system, running our algorithms inside this secure setting. But the plan with Health Data Research UK is to do that much more generally across health records data, population health data. There's projects at various sort of levels, but there's something called the Health Data Research Alliance, which is trying to bring all of those data owners at least into a common framework, so you can start doing analysis in these different environments and aggregate the results together. You don't move any of the data itself. You try and set up a system where you can federate between trusted environments. And there's been engagement with public and patients around that. And it seems people are much more relaxed if there's proper oversight, if there's patient representation involved in the review of projects that carried out, and there's transparency.
It says a lot about our society if there's not a lot of people volunteering their DNA for science, but then they have like, ooh, am I 1% in any of any ancestors? So that does say that maybe to make it as a test. Say, volunteer and we'll tell you if you are, 10% hamster.
You're suggesting that people are more willing to give up their DNA to private companies who will give them a dodgy readout of what race or ethnicity they are compared to an NHS-related health data project. Is that what you're suggesting?
That's it. I'm just saying, right? We all know someone that would much quicker go for I'm gonna see if I'm 10% rabbit than if I or help research. So.
Well, that is an interesting point. I mean, that there obviously are a lot of valid privacy concerns around sharing patient data, whether it's on illnesses that people have had, NHS hospital data, or of course genomics, genetic data. And you were just talking a bit there about Health Data Research UK. So I don't know if, can you tell us any more about what's going on? You mentioned the specific kind of research computing environments that are allowing for this kind of privacy preserving research to take place. My understanding is that what this kind of thing allows you to do is it allows scientists to essentially work on this kind of sensitive personal medical data and do whatever like cool science they want to do on it. But the point is it's in a kind of computing environment that's not exposed to the internet. It can't be hacked into. It can't be, the people involved in it can't easily, share stuff out of it. What are the other sorts of things that enable that kind of research to take place safely?
So, there are other things about, firstly, the data, even if it's in the secure environment, is all de-identified. So you can't work out automatically who somebody is. And because you control who can log in, they've all had to sign legal agreements saying they're not going to try and re-identify anybody. And some issues where data has been anonymized data has been outside and some people have tried to re-identify. Normally you need external data for that. So by controlling data that goes in as well, you put additional controls on what happens. And then, of course, you also have oversight. People have to propose what projects they're going to do. So those have to be viewed and vetted by access control committees too. So there's quite a lot of wrapping around researchers. So a lot of it's around confidence and transparency. So, Genomics England has a large participant panel who, are updated about what's going on and are on these committees. And there's lay summaries of the projects that are being carried out inside the environment, both academic ones and company ones as well, because companies can do exactly the same thing. They can come and do analytics to look for targets for new drugs, for example, but they follow exactly the same rules. They can't take the data away. They can only export summaries.
Is that kind of approach, both in terms of the software that enables it and the rules you mentioned, is that something that sort of Health Data Research UK is pioneering? And is it going to become? I can imagine it's going to become much more of a thing, much more frequently a used approach as we enter an even bigger world of health data, whether it's proteomics, as we discussed, coming out of DeepMind or whatever else it may be. There's going to be more big data sets in the future.
Yeah, so I think that is part of the point, that data sets got very large, and so it's actually quite hard to move these data sets around, even if you were allowed to. And similarly, computing has evolved, so it's easier to put your algorithm into a container and move that container to the place where the data is and run it there. So this is an evolution of computing standards. And storage costs for very large data sets, it's expensive. You actually don't want to have lots of different universities or companies all having to build very big storage facilities to hold data that maybe they're only going to analyze a small number of times. It's much more efficient to have it in a single place, and then you can do the security very carefully. And you can also extend this into the cloud, because the cloud providers have worked out how to do security where everything's encrypted and they can't look at the data either, because you, as the controller of the data, hold the keys. So the security, 5 years ago, this probably wouldn't have been possible, but the security design has now sufficiently advanced where that's possible. And then that means that if you want to do some very sophisticated AI, for example, involving a lot of computers, you can actually do that in this sort of secure cloud framework. So Health Data Research UK is certainly, trying to coordinate and lead on the standardization. And so, in fact, we've published a green paper on trusted research environments. And that's, the word TREs actually is being mentioned by quite a lot of different groups. NHS England, NHS Digital has been setting up a TRE as a way of, allowing analysis across some of their summary health data sets in a secure way.
I was going to say that is a reassurance to know that there's so many safety steps at any point to protect the data and the people. But you were going to ask something, Ed.
No, go on. I was.
It's always a bit of an awkward moment when we're just. So who talks now? Sorry. I forgot what I was going to say.
Do you want to go to next question 5? I guess we kind of already asked about Genomics England.
Oh, yes, we can go to question 5. Because you mentioned, Tim, you mentioned a little bit about sequencing 100,000 genomes in the NHS and how that was a goal, and it was a goal that was met in 2018. So you already said a little bit about what comes next. So do you want to expand a bit on what's the next goal and what's the idea now after that?
So the Genomics England project was to sequence 100,000 genomes in the health service for clinical care, but also set things up in a way where those 100,000 genomes could also be accessible in a secure environment to do research. But the key point was to get to show, demonstrate that it's worth sequencing, it's health effective to sequence whole genomes for at least a subset of conditions. And so alongside the sequencing and providing individual reports that have gone back to the NHS for each of those patient families, there's been a health economics analysis around, what's the benefit in terms of healthcare for those families in terms of the NHS. And so rare diseases and some cancers, it's been established that that's health effective. And so that's led to the NHS deciding, this was 100,000 was a special project. This is now a permanent feature of the health service, launched as the Genome Medicine Service. In many ways, it sets up a single testing directory, which includes, the old tests, the original single gene tests, which have existed, panel tests in the health service for different conditions, supplemented by whole genome sequencing where that's appropriate, where that has sufficient clinical value. And of course, as we expand our knowledge of interpreting genomes, the number of conditions where it's worth doing a whole genome sequence will increase. And so, costs will go down, our experience will increase, and you'll see more and more people that have a stored genome. Because, of course, you sequence a genome, it's not like a normal test in the health service where, you store the result in a record system and don't necessarily keep the data. A genome doesn't change during your lifetime. If you get cancer, the cancer genome is different, but your germline genome isn't going to change. And so if you're ill for something else, you can go back and look at the genome from another point of view. If you've got a stored genome, potentially the GP systems could query that genome and see which drug is most likely to work for you, because we know quite a bit about pharmacogenomics, but we don't test for that because you wouldn't want to wait for a test result to decide, A or B drug. We know these two drugs are perfectly safe. We just don't know which is optimal for you. But if you've got a stored genome, you can do that electronic lookup instantly. And so you can build that into the electronic health record system. So there's quite a lot of future engineering having got to this point. We're doing the low-hanging fruit to improve health service, but you can see quite a few other things. With our knowledge we have now, we can start plugging those systems together. And this is the first health system in the world that has deployed this. No other country has managed to do this so far, other than the UK.
I guess this is because, as you said, it people could see this as they do this test once, and then it's not like, as you said, a test that needs to be repeated. So, do you think it would be that one of the goals is to implement it and just do it for everyone, like a standard checkup test that you have to do when you get up and to a certain, I imagine a certain age. You have to.
Do you mean when you say test B? Do you mean getting the genome sequence?
Yeah. Secret. Yeah. Sorry.
So the point is that at the moment, unless you're ill and we need to diagnose something and a genome is going to be helpful, we don't understand enough really to make it worthwhile. Because, this is what these companies, providing tests for, they're saying that they can give you some risk score, but it's just a risk score. And we don't understand enough in most cases for that really to help your health. So at the moment, it's just being used in cases where we know it's health effective, rare diseases and some cancers. And that list will expand over time. But we can't really do prediction, basically, at the moment. Well,
it's interesting you say that, though, because it seems to me like, as you said, as you sort of preempted there, that we might be able to in the future. And this is something that if the project was 100,000 genomes of, people in the UK, and it's still ongoing with more, it does seem like what B was getting at there of eventually having everyone's genome, everyone's readout of their DNA on record. There's got to be science that you can do there, and as you've explained, there are safe, privacy preserving ways to do that science now. So do you not see that eventually we will get to that point where everyone has their DNA sequenced at some point?
I think it's all, from a health service point of view, it's a question of, when is it health effective to do that? What number of people? And that, the fraction of people where it's useful for their health will increase over time. But, it's actually building a system to handle 100,000 genomes is quite challenging. And we are going to scale it up to 1 million over 5 years, because there'll be 500,000 done in the health service, and there'll be 500,000 as the cohort UK Biobank, that's a set of volunteers. So they're not going to get feedback individually because it's a volunteer cohort and it's a research cohort. It's not set up for clinical return, but it will be another 500,000 genomes. So we'll have 1 million in the UK. European Union's talking about doing 1 million across Europe. If we can work, information will get shared, statistics will get shared between these different cohorts, and that will gradually increase our knowledge and allow us to use it for more things. And of course, the protein, being able to predict protein folding, I think removes a massive bottleneck in our ability to interpret
and to get useful information out of those genomes. Yeah. I think the way you've described it kind of makes sense, because I think what B and I were imagining, it was like, well, why not, if we know it's going to be useful in the future, why not have,
everyone.
Why not have a, government commitment to sequence everyone's genomes? But actually, what you're saying is to advance the number of people who are having their DNA sequence on the basis of, well, this cohort of people, it would be useful to know for this, learning about this particular disease, and so on and so forth. We're going to get more and more done over time, and if we're focusing on the most important people to do that for first. So, and given that it's not easy to just quickly do it for everyone now, that does make a lot of sense.
Yeah, and it is, cohorts for doing research problems, and then the testing directory to decide the clinical reason for doing it. So those, people frequently ask the 100,000 genomes, what was the target of the research? There wasn't it wasn't designed, it wasn't a research study. It was based on unmet clinical need. The people where the genome would most help had a highest chance of producing a useful diagnosis. Those are the ones that got sequenced. So there was no bias across the whole country, really, in terms of access to this.
I was already thinking about all of the sci-fi options that we had and how far we are from them. and then that everyone could have like their fingerprint in the system and their genome, and that would be, but then I do imagine that it would have to be such a big, storage for everyone's genome to be saved.
That's one consideration that amongst many. Yeah.
So yeah, Genomics England 20 petabytes for 100,000. We're cruising at 60 petabytes now.
Oh, so
sounds like
no
a
biggie.
lot.
It's not a small amount of data.
Okay, yeah. So I did have one related question there, which was, I think I saw that there's a push from Genomics England to sequence genomes of people who have had COVID. Is that correct?
Yeah. So that's obviously COVID has changed a lot of things. That's affected Health Data Research UK. There's been a big focus about using these national data sets. In fact, this is the first use of the NHS digital trusted research environment has been around COVID. Genomics England angle, of course, is genomes. And so it's part of a national alliance based out of Edinburgh to identify those patients who were most affected by COVID, and similarly those who were infected and least affected, and then sequence both of those and do comparisons. And so that's ongoing. In fact, because of the early phase, after lockdown, the numbers went down. Actually, took a while to get enough patients, but that they're being sequenced, and the target is 35,000 whole genomes, 20,000 most affected and 15,000 least affected. And then that's also, the data is also being linked to international data sets, because you just need large numbers to have the statistical power to find parts of the genome that are most significant.
Yeah, interesting to point out, I guess, what's being thought of here, because obviously most of our listeners will think, well, COVID-19, it's caused by a virus. So, but what we're talking about, I guess, is people's susceptibility to having,
people's susceptibility
to having the worst case of the disease, if they did get infected. And what you've just said there is that you're taking people who have had it, but there's one subpopulation of those people who had it really badly, and the other being ones who didn't have it badly at all, to see what the differences are. Cool.
Which makes sense, because even with the protein folding even can be helpful, because what if one of the proteins folds makes someone less susceptible to have a lot of symptoms rather than just having it normally?
Some difference in their protein structures allows the virus to infect better, you know.
Well, so that's the spike protein, and that's been one of the CASP targets this year. In fact, there was a special subcategory of CASP, specifically for COVID. And of course, the DeepMind models have turned out to match very well because there was obviously a rush to try and do X-ray crystallography on these as well, but not all of them have been crystallized. So I think the view is that now that people should start assuming the DeepMind models are correct, in cases where there's no X-ray structure.
Wow, that's a vote of confidence, definitely.
And it can also help if we can assume that it's correct. It's also, imagine, let's hope not, but there's other pandemic and there's other emergency. Hopefully after this one ends and we have some years of peace. So because in that case, we wouldn't have to necessarily wait the entire time for the crystallography. We could just have a prediction and start working on solutions earlier, I suppose.
Yes, exactly. So, think of all the different pathogenic organisms that are out there where pharma is looking for targets to develop drugs. And one of the big costs is frequently, where's my target? What's its structure? If you can take a genome, which of course is now very easy to sequence, and predict all the structures, you're in a much better position to then work out where are good targets. So it can revolutionize, particularly in those neglected diseases where there isn't as much money. It should really help with that. In fact, DeepMind have said that they would like to target some of those.
The not so cool diseases that have less funding.
Right. Well, that's important too. Just to decode some of the biology jargon that I noticed you used there. So when you're talking about targets, you're talking about the proteins that are on a disease-causing virus or bacteria that drug manufacturers might want to target with the drugs they develop. So it would be, the drug in some way interacts with that particular protein on the virus or bacteria.
Yes, I mean, most drugs work on a protein target. Not all by any means, but it's one of the classic. So if you know the pathways within a particular organism, you try and target things that are critical to that particular organism with a drug that's going to knock some pathway out. And obviously you need the structure for that. And you can do screening, of course, high throughput screening of compounds, but it helps if you've got a structure as well.
That leads me on quite nicely to the next question, which is, as well as sort of drugs for treatment, of course, one of the big things that's ongoing at the moment is all of the developments of vaccines for COVID-19. And of course, as many people will have heard in the news, not just us, people who've studied biology, people will be quite familiar with this now, that the way that vaccines also work tends to be, inducing the immune system to create antibodies, which themselves latch onto these targets, which you call them antigens. But we've got multiple vaccines that appear to be effective in preventing the disease now that have been developed by lots of different companies in different countries. Do you have any opinions about all of this at the moment? And I'd be interested to hear if you have any thoughts on the particular one that's being rolled out in the UK, which is the Pfizer vaccine, which is interesting to us as biologists because it's the first vaccine based on RNA. Do you feel generally optimistic or pessimistic about where things are going?
Yeah, I think that, for example, for a long time, there have been unsuccessful attempts to develop vaccines against various things like AIDS, for example. And this time around, there's been a coordinated effort worldwide. A lot of different techniques have been applied. In some ways, actually, you could say this is a bit like CASP, a coordinated effort with lots of different people trying different approaches. And some of those have turned out to be successful in taken the science forward through this cumulative effort, this collective effort.
That's an interesting thing to pick up on. So the thing that's impressed you most has been the fact that it has been a sort of a natural competition, if you will, that lots of people have risen to the challenge. I felt impressed by the global scientific community. I mean, there's not been vaccines developed this fast before, so.
No, exactly. And a lot of virologists were really quite pessimistic, saying, we know this class of viruses and we haven't had much luck previously with this class. And so, this push to do something has seemingly pushed the field forward, which is, very, very encouraging, especially with the timescales. I mean, it's had all kinds of effects. It has, there's been a real push in terms of sorting out some of the data things inside HDR UK or through the alliance that HDR UK has put together. And that's helped to drive, some of the federation and, enabling analysis that we'll be able to use in other settings later. Right.
Yeah, so I think it is it's worth pointing out that although the people who we're most excited about in the news are the people who developed the vaccines, but the data science community has sort of risen to the challenge of COVID-19 as well.
It's like data scientists everywhere are saying, no, we also did things.
All right. Okay. So I think on that note, Tim, it's a good point to end with a bit of a bonus question, which hopefully this is more in your area of expertise than the last thing I asked. But so it's a bit of an open-ended one, but it's very relevant to everything we've been discussing so far. Just how many years or decades would you say we are from being in the era of truly personalized medicine? And I'll let you interpret that as you will.
So that's what I think. I mean, that is what Genomics England's doing. It's, people are affected by genetic conditions. That's their genome has affected their health. And so one step away from that is, what other things can you do maybe preemptively? Now, it's complicated, it's a very complicated system, and then there's environment as well. It's not all genetics by any means. But there clearly is some information in there that can be useful in terms of preemptively spotting problems. We just have to get better at interpreting it. And as I said, having all the proteins is going to help in that process. So, the most obvious cases are things like pharmacogenomics, where it does look like there's some variant in around 80% of people that we've sequenced so far that would affect, how they react to some drug, which is fairly common. If you look at it, it doesn't mean to say they'll ever be given that drug, but you can see enough patterns that if you had a genome, you could probably improve healthcare overall over the population.
Maybe avoid all of those allergic reactions,
side effects.
And
Yeah, exactly. And you, in terms of personalized medicine, there is a community of health systems that have some sort of genome medicine activity. One of those is Thailand, because they have, 10% of people have a particular subtype of immune system, HLA, which gives a particularly severe reaction to skin condition. And so if Stevens-Johnson syndrome, it's very, very serious. If you treat that and you didn't realize that they had that susceptibility, it can cause death. And so it's worth them testing before they give drugs in that context. Fantastic.
So in other words, we're kind of already at least at the beginning of the personalized medicine era.
Yeah, the future's already here. It's just not evenly distributed.
I was going to say, it's scary to think as well that this is what we know. Imagine what other things exist that are much more advanced that we don't know yet. So that's just, sci-fi.
Scary but exciting. So
yes,
to think that
it's definitely exciting.
We don't have hover cars yet, but I guess that's fine.
Well, I think I'd rather live in a world where we can have our genetic diseases and cancers treated than in the world of hover cars. But
exactly.
Hopefully we can have both. There's no reason why not. On that note, Tim, thanks very much for coming on the podcast. Before we let you go, where can people find out more about your work, and do you have social media that people can follow?
So I'm on Twitter as TimJPH. I actually do have a blog on Medium which literally I've only done a couple of posts, but one particularly around the AlphaGo DeepMind AI thing. And of course you can find me at King's, on the websites there.
Thank you.
All right. Yeah. Thanks very much.
Thanks for having me. It's great.
If you have an interesting topic you'd like featured on the show, a guest recommendation or a burning question, email podcast@turing.ac.uk.
The Turing Podcast is hosted by Ed Cowstry, B. Costa Gomez and Joe Dungate, and produced by Dan Whitfield for the Alan Turing Institute.
Music for the podcast was provided by Jam and Sun. You can check out his latest releases at jamandsun.bandcamp.com.